This is the lung of a 60-year-old man who has been smoking habit for more than 30 years. Like all other smokers, his lungs is filled with powerful mutagenic substances, some of which are neutralized by the liver, which pass through the kidney and enter the urine. The powerful substance which is left in after the urine, the substance begin to attack the cells lining his bladder and damages the cell to enter inside. And based on changes from the substance or tar, the genetic mutation undergo a changes and turned into an active oncogene, and this deteriorated cell began to react crazily, which instantly becomes active. This is a gene mutation resulting in an active environment and becomes an immortal demon in human body. Day after day, year after year, this oncogene cells reproduce billions of cancer cells, which eventually grow into deadly tumor. Cancer has become the leading killer of human life. In 2022, an estimated 9.7 million people died from cancer worldwide, which is about 1 in 6 deaths. This number is expected to increase to 15.3 million by 2040. Yet, even though cancer is the leading cause of human death, many people, including those diagnosed with cancer, know nothing about it. What exactly is cancer? Why do humans get cancer? How do cancer cells form tumor and take human lives? Modern medical research on cancer can be tracked back to more than 200 years ago in 1775, when London Dr. Percivall Pott, English surgeon, observed a strange phenomenon. The occurrence of cancer seemed to be related to the patient's living environment and lifestyle. In a paper, Pott described how the London chimney sweeps develop scrotal cancer at much higher rates than the rest of the population. Subsequently, increasing number of reports strengthened Pott's view that people in harsh environments were more prone to cancer. Like all mines in eastern Germany, miners or coal workers are more likely to die from lung cancer. People working with X-rays were found to develop skin cancer and leukemia. Since the 1950s, the lung cancer rate among smokers has risen significantly, 20 to 30 times higher than that of non-smokers. In 1915, researchers Yamagiwa Katsuzburo and Ichikawa Kichi from Hokkaido University in Japan first confirmed that chemical substances can cause cancer. They repeatedly applied coal tar to the ears of rabbits, causing skin cancer to grow. This finding strongly suggested that chemicals are a major cause of cancer. One example is from Zhejiang, where a farmer discovered that after rain, certain fungi on rice crops would secrete carcinogens, leading to high cancer rates among locals who consume the rice. Similarly, in some hometowns with chemical factories, cancer incidence is significantly higher among residents. Miners inhaling dust in new mines also have a very high rate of lung cancer. But how do X-rays and chemicals turn normal cells into cancer cells? What is the fatal blow? Over the past century,
well, molecular biology has done some incredibly important things by introducing us to the basic concepts by which cancers arise from normal cells as a result of mutations and other changes in the way our genes work, and by allowing us to understand how cancer cells interact with other cells in the body, the cells that make blood vessels, the cells that make the tissue that surrounds a cancer, the cells that comprise the immune system that is sometimes capable of rejecting a cancer.
In the mid-19th century, Austrian monk Gregor Mendel discovered through pea-plant hybridization experiments that there are basic factors a genetic characteristic will be passed from the generations though this basic factor within organisms that pass on hereditary traits, which are now known as genes. In 1953, molecular biologists James Watson and Francis Crick at the Cavendish Laboratory at the University of Cambridge proposed the double helix structure of DNA. This opened the door to understanding the fundamental structure of life. Genes are specific sequences of nucleotides on DNA or RNA molecules. Each DNA molecule is a double helix consisting of two mutually coiled strands. Each strand is composed of 4 bases, randomly arranged, composed of long chains. The entire information content of the human genome is composed of a DNA sequence of 3 billion base pairs of DNA sequences, divided into regions representing 70,000 to 100,000 different genes. DNA with specific structures within these regions control the synthesis of corresponding proteins, forming various cells in our bodies that perform different functions. After the 1970s, researchers like Harold Varmus and Michael Bishop from the University of California, San Francisco, conducted experiments on animal cells. More than 20 genes from animal cells were isolated, including avian myeloid tumor, rise from mouse sarcoma virus, and feline sarcoma virus were discovered. Once these genes are captured by viruses called retroviruses. These genes, termed proto-oncogenes, are present in a wide range of vertebrates.
We now know that there are hundreds of different genes out of the 21,000 genes in our body that can play a role in the development of a cancer. And when those genes were first beginning to come to light, it was apparent that some of them worked as cancer genes because mutations had made them stronger and they worked in the presence of a normal gene.
It turns out that these proto-oncogenes naturally exist within our bodies and they are normal components of our bodies. This crucial discovery confirmed that cancer is a disease closely related to genetics. Through the scientific research, the causes of cancer have gradually become clear. A chemical right factor or retrovirus invades cells and they attack a key proto-oncogene and transforming it into an oncogene. Driven by the oncogene, the cell begins to grow relentlessly, eventually forming a lethal tumor. However, the real cause of cancer is far from simple. Medical scientists soon raised questions. If a single cell mutation causes cancer, in a person's life the probability of such a mutation occurring should be equal. However, epidemiologist statistics tell a different story. A 20-year-old has twice the risk of developing cancer compared to a 10-year-old, and at 70, the risk is 700 times higher. As the age of the surveyed population increases, the incidence of tumors rises sharply. This indicates that the older you are, the higher the chances of cancer. This shows that cancer does not result from a single cell mutation but goes through multiple steps. In 1969, Henry Harris of Oxford University compared normal cells with cancer cells and injected them into mouse. And he was surprised to find that none of the mice developed cancer. This shows that normal cells have another kind of gene that can suppress cancer. This discovery opened a new door for cancer research. A door the last decade of the 20th century. More than a dozen tumor suppressor genes were discovered one after another. The most famous being the p53 gene first tracked by Lionel Crawford at Princeton University. The p53 gene controls the life and death of cells. Once DNA mutate under external stimulation, p53 repairs the DNA. When cells are controlled by cancer genes and begin to grow uncontrollably, p53 gene will induce the death of cancer cells. Among the more than 100 types of cancers currently known to humans, over 60% of human cancer is caused by the loss of the p53 gene in cells. Other genes contribute to cancer when they are lost. we now call it tumor suppressor genes, is lost, it can lead to a major disaster. Let's take the p53 gene as an example. It is located on the 17th chromosome. Since the chromosome is a double helix structure, with each p53 gene has another copy of the gene, and they are located on both sides of the double helix. Their DNA sequence are compared and genetic information is exchanged. For every million cells, one cell's tumor suppressor gene will be mutated, and this mutated p53 gene will be repaired by the p52 counterpart on the other end of helix. But in 1000 cell divisions, one cell will have a mutated version of the p53 gene completely replacing its counterpart. At this point, at the time both p53 genes in the cell have been lost, and the cell is on the verge of losing control. Copies of the gene are delicate, and any errors during cell division can lead to cancer. Hence, the probability of cancer arising is very high. Because our cells are changing from 1 to 2 and any time an DNA error occurs, it may be the cause of cancer. So speaking of it, there is very high probability that the cell that has lost p53 gene will begin to divide and reproduce, and none of its children's cells will have the p53 gene if a chemical mutation activates the proto-oncogene of a certain cell in this batch of cells. A cancer cell will finally appear. Since our body is constantly classifying and reproducing, so this is why both the gene breaks, tumor suppressor genes, and the accelerator, proto-oncogenes, malfunction helps cancer to develop. In fact, it is the common result of proto-oncogene mutations and tumor suppressor gene loss. In cell molecular biology at the level of human beings, for the first time scientifically explained the cause cancers. But how proto-oncogenes mutate into oncogenes and why tumor suppressor genes are lost still remains an unsolved mystery. The study of cellular molecular will also provide future cancer treatments. Injecting missing tumor suppressor genes back into cancer cells to prevent excessive growth and induce cancer cell death is a future treatment approach. This idea is one of the important means of cancer is not science fiction but a feasible direction already supported by some practices. How can we find a way to restore the inactivated tumor suppressor gene in the seed to its normal function, to make it work again, and to prevent occurrence of this tumor and development? A cell has to experience the loss of a tumor suppressor gene, p53, at the right time. It activates the proto-oncogene and turns into cancer cells. This chance is as difficult as winning the lottery jackpot. However, within a human lifespan of 70 years or more, human body will produce 48 billion to 68 billion cells. Given such a large number of cells, the probability of developing a cancer cell becomes significant. Before liberation, cancer was rarely heard of, on the one hand partly due to undeveloped medicine and on the other hand partly because the average lifespan was under 40 years. Today, with increasing life expectancy, world is gradually entering an aging society, and the number of cancer patients will also increase. However, the emergence of cancer cells does not necessarily mean that it will grow into a deadly tumor. Let's return to the microscopic world to see how cancer takes over human lives. When a tumor cell grows to a diameter of 1 millimeter, molecular diffusion cannot provide sufficient nutrients and oxygen to the cell group. Without oxygen, waste cannot be cleared quickly, and cells start to starve and suffocate, leading to cell death, which lead to metabolically poison, which causes mass death of cells. Therefore, the volume of tumor cells grow for years or even decades, and that results into the tumor growth before it exceeds the 1 millimeter diameter. The cancer cells group figures out way to increase the capillaries inside. Some cancer cells mimic surrounding normal cells and secrete growth factors to attract endothelial cells from nearby tissues, creating capillaries that penetrate the cancer cell cluster, providing more nutrient-rich blood. Now the cell cluster can rapidly multiply, achieving their long-held goal of expansion. cancer cell numbers start to break through. Normal cells do not overexpand, but mutated cancer cells increase significantly in number, forming a tumor. When a tumor reaches a diameter of 1 centimeter, it contains around 1 billion cells. But even at this size, it does not necessarily cause death. Among the cancer victims, less than 10% of cancer deaths are due to primary tumors, and about 90% die from cancer metastasis. Some cancer cells use blood vessels as an migration route, while others cancer cells choose the lymphatic vessels. They drift along these tubes, land on other parts of the human body. These adventurers are almost faced with near certain disaster. They must survive the stormy wave circulatory system and adapt and try to grow in an environment that is rejected by all hostile environments. However, the tumor has already developed significantly and creating numerous cancer cells in human body. Eventually, some establish new location in human body. As the tumor grows, and when this tumor grows to a certain point where its nutrients and space becomes inadequate, it starts driving it to expand elsewhere, like in body, like humans exploring new frontiers. Eventually, these metastatic lesions grow into deadly tumors. They begin to absorb large amounts of nutrients from human tissues, causing normal physiological dysfunctions, weakening the immune system, causing organ failure, and leading to various complications. At this point, the shadow of death hangs over cancer patients. Today, in the 21st century, people have deep understanding of cancer's pathogenesis. After 4,000 years of battling cancer, mankind is gradually overcoming from an era of inexplicable fear and can now approach cancer treatment rationally and optimistically. Speaking from people all over the world, as global life expectancy increases, the incidence of cancer correlates directly with longevity. In the past, an average lifespan of over 40 years meant cancer was not a significant issue. Early deaths from infectious diseases and poor living conditions left little time for cancer cells to develop. But now, living into the 70s and 80s, cancer incidence is directly related to average lifespan. If we put it this way, the probability of this cancer occurring is directly to our average lifespan. The relevant of relationship is that when our average life expectancy approaches 100 years in future, the incidence of cancer rate is likely to be at least 50%. In 2006, the World Health Organization officially declared cancer to be a type of chronic disease.
Well, it's a chronic disease because it doesn't kill people quickly. It grows relatively slowly, and in general, when a person is diagnosed with cancer, there are several, one has quite a bit of time to make decisions about treatment. Moreover, even when treatment is not ultimately successful and patients succumb to their disease, it's still, there is a long time course for treatment.
From this perspective, it is also a chronic disease. According to the World Health Organization, WHO, between 30–50% of cancer deaths could be prevented by avoiding or modifying risk factors and using evidence-based prevention strategies. A 2020 study led by researchers from the WHO's International Agency for Research on Cancer, IRC, estimated that 68% of premature cancer deaths globally were preventable. Some lifestyle changes that may help reduce the risk of cancer include maintaining a healthy weight, exercising regularly, not smoking or using smokeless tobacco, eating a healthy diet, limiting alcohol, protecting yourself from the sun and avoiding tanning beds, protecting yourself from sexually transmitted infections, getting screening tests regularly. Worldwide, eliminating smoking could reduce lung cancer incidence by 22%. Preventing Helicobacter pylori, hepatitis B, hepatitis C, and papillomavirus are related to chronic infection, which may cause gastric cancer, liver cancer, and cervical cancer respectively. These 4 infections and eliminating smoking could reduce cancer cases by 29%.
Well, despite the fact that people always think about our need to cure cancers, it's obviously much better from the patient's point of view never to have had cancer at all. And yet there are not a lot of people running around saying, my cancer was prevented, because they don't know that it was prevented.
It can be seen that before humans have completely conquered cancer, cancer is a disease that mainly focuses on prevention. In addition, even if we do get cancer, we don't have to be too afraid, because the growth and spread of tumors is a very long process. Medical experts in the world suggest that every treatment is more likely to be completely cured. Modern medical research has given humans a very in-depth understanding of cancer. Medical experts believe that in the 21st century, humans will make breakthroughs in cancer treatment, possibly curing it with a single vaccine injection. However, today, if a person makes up his mind and starts to change his living habits, it is possible to avoid occurrence of cancer. Even if unavoidable, the mortality rate can be reduced to as low as 100% through treatments like surgery, chemotherapy, and radiation. The lowest point and the key to all this is whether we can actively publicize the pathology of cancer to the whole world and even everyone in a scientific way. In this world, nothing is more important than saving the lives of others and maintain own health. The key lies in educating the public about cancer pathology and promoting health awareness. There is nothing more important in this world than saving lives and maintaining health. If the information provided in this video adds value to your knowledge, please hit the like button and support us by subscribing to our channel for creating more informative content for you. Thank you for watching. See you in next informative video.